Today we're going to be talking about the synapse, right? And that generally it falls under the general category of cell communication, right? One cell trying to communicate with another cell. And we had already talked about the endocrine system, where you had those secreting cells in certain glands, where secreting out through exocytosis, right? The hormones into the blood. They travel around the blood, leave the bloodstream, and then find their target cell, because those target cells had a specific receptor for that hormone, right? So they go all around the body, and stuff that was released in your pituitary gland would just circulate all the blood vessels in your body, right? Which is going to reach all the tissues in your body. And the ones with the proper receptors would become activated, right? And the activation effect might have been to maybe insert glucose channels, right, in the cell membrane or something like that, or increase protein synthesis, something like that, right? But in general, like cell communication, you're just secreting some kind of molecule. Well, unless you're directly attached to another cell, like epithelial cells or something, they might be directly attached and they can communicate this way. Other cells can communicate with cells a little longer distance by secreting out enzymes, not so much in the bloodstream, but in a similar way, you secrete it into the interstitial fluid, and then it would reach your particular target cell, whatever had the receptors, right? This is what happens during any development of an embryo, right? Cells act as signaling centers and they turn cells into other things. They activate gene programs to make your head or your feet or whatever, right? Your particular organs of your body. So that's how cells communicate with each other. They send out these signals if they're not directly attached to each other. Another way, at some point in evolution, right? This is happening. If you're an amoeba in a pond, you can do this and communicate with your fellow amoebas, right? Or your bacteria or something like that, right? You can spread out this signal and other cells can detect it and respond accordingly. But let's say you're this cell and you want to only communicate with this cell, even though these cells might have a certain receptor. So you could maybe, instead of just putting it out into the surrounding whatever medium, right? Maybe you can grow a process and then talk to him directly right over here, right? And then even if this guy's did have a receptor, right, it wouldn't matter, right? You could talk to this one and only this one, or maybe branch off and talk to this guy as well, right? Or this guy, right? You can go anywhere. That's basically what's happening with neurons, right? They're sending out these long processes, the axons, which we learned about, to specifically talk to other cells and only those cells, right? So that's really what your nervous system's all about versus your endocrine. So that connection in between that extension, okay, again, the axon leaving that neuron is going to communicate either on the dendrites or the cell body or sometimes the axon. They're going to communicate through this structure called a synapse. And that's the region where the communication occurs between those two neurons, or as we saw with the neuromuscular junction, a neuron and its effectors. Or, as we'll see with sensory stuff, a receptor and a neuron, right? So any of these will involve a synapse, right? The connection between those terms. So there'll be a pre-synaptic neuron, the one that's sending the signal. The one that's sending the signal is the presynaptic. The one that receives the signal is the postsynaptic cell. Usually we'll be talking about neurons, but there's also the effector cells, the muscle, right? So in the case of our neuromuscular junction, the postsynaptic cell was the effector cells that received that signal. Right? So that's your synapse right there. And the terms we'll be using: presynaptic, postsynaptic. We'll be mostly talking about this chemical synapse. In fact, we'll only be talking about the chemical synapse after I discuss this very briefly, but electrical synapses, this is connection in between two cells. and they're going to have a direct connection through these gap junctions. And we already saw these a little bit when we talked about cardiac muscle cells and smooth muscle cells. They have these gap junctions that can pass an electric signal from one end to another. Right? So one cell is directly connected, and ions, charged ions flowing through here is going to come down the axon, do that action potential, and then spill in here, right? And then cause this graded potential, right, based on that. And that'll be pretty much instantaneously, as we see with cardiac muscle cells, right? They're all kind of connected to each other and all firing it. This is, you'll see that when you get into Bio 145, especially with your cardiac muscles and maybe a little bit of your smooth muscles, these gap junctions and passing electrical signals. And, you know, you could act as a single unit very fast, right? Very, no delay in between the time the action potential reaches this cell and the time this gets the effect right there. Right, so electrical signal, electrical signal. On the other hand, most of what we'll be talking about, most of what's occurring in the brain is this chemical synapse. That is, you're going to have this action potential come down the axon, reach the terminal bouton, the end plate right here, and secrete a chemical signal, the neurotransmitter, which is going to diffuse across the cleft right here and bind to receptors. They're going to open up ion channels, usually of some sort or another eventually, and then cause that graded potential as we talked about last time. And you'll notice there's a little bit of a delay right here, right? It's going to, the action potential comes down, then it takes time for this vesicle release, the vesicle diffusion, the binding, the opening of the channel. I mean, it's only 0.5 milliseconds, right? Which, you know, is 0.5 thousandths of a second, which is very fast, but there's still a little bit of a delay right there, right? In between this action potential and the graded potential that the postsynaptic cell receives right there. So why would you even do that, right? Why would you have this chemical signal? This is so much faster, right? Because sometimes it turns out you want that little delay. It's going to give you a lot of more variability in how you can pass messages along to the next cell. But basically, we're going to go from this electrical signal, graded and action potential, then go to a chemical signal. And then there's going to be a graded signal, and then an action potential, then a neurotransmitter release, the chemical signal, graded potential, action potential, and so on, right? This will all be happening at the synapse right there, that chemical synapse. So here's, you know, when we get into the pathways in the brain, this is what you always want to remind yourself of. Keep everything in context, relate it to everything else you're learning about the nervous system, right? Because here is one of those neurons, or here's two of those neurons really, and they are picking up information from the environment, right? Either touch or pain or something like that. And then they're going to bring it into the central nervous system, right? They're picking up in the dendrites. And this was some kind of chemical signal or direct physical stimuli that caused this action potential down the sensory neurons. And they brought that action potential all the way into the nervous system. And we'll follow this fine touch one for a second here. It goes in, action potentials is going to go up here, and then it's going to talk to another neuron in your brainstem for some reason. And it's just going to stop, pass that signal on, and then that one's going to go up. That's going to cause a graded potential if it's strong enough, and it's going to cause an action potential up, and it's going to stop in this region called the thalamus, as we will learn. Right, and from there, this is like a relay station that's going to send it up to the brain, right? Another chemical signal. If it fires, it's going to go up here and tell your brain that you had some kind of touch, right? So you had two, right, relay neurons in between that actual touch and your perception of that somehow. In fact, by the time you get to a perception, there's probably been like a couple of hundred synapses up in your brain right here. But why would you stop, right? If this is going to slow things down, don't you want to know instantly that a bug is crawling on you, or you're in pain in this case? There's a pain pathway, temperature pathway, This neuron comes in, it connects to a neuron right in the spinal cord. That one goes up. There's only one in this case, only one synapse that stops in the thalamus, and that's also routed to your brain. Your brain eventually will know that you have some kind of pain or heat in wherever that neuron came from. Let's say your thumb or something like that, right? So again, why would you want to have these synapses, right? In this rate, why don't you just go right up to the brain? Again, sometimes you don't need to know everything that's going on, every single stimuli in your body. So right now, if you're sitting down, there is pressure on the back of your thigh. Right? Can you feel that? You can. There's also noise somewhere right now, somewhere in the background right there. If you listen, you could hear it. Maybe a clock ticking, maybe somebody screaming in the background. Right? You could hear that stuff. You weren't thinking about it a second ago. All that information was coming into your body right there, but you weren't paying attention to it. Or sometimes you're asleep and nothing gets really through to your upper brain right there, right? So all these are going to be like modulatory, right? They're going to allow you to kind of modify that information, right? Maybe just totally ignore it, like the back of your thigh a second ago, until you were conscious of it, and then you could redirect it, and then you could notice it. But there's all sorts of ways that you don't need to know everything, right? And this is like a filtering station that's going to kind of bring in the information that's important to you and ignore the stuff that isn't. So you could do that at these synapses in a way that you couldn't do with your electrical synapses that would go right up there, right? Okay, so that's why you might have these junctions and different relay neurons in your sensory pathway. Again, we'll get into this. You don't have to know this right now, but you know, all these synapses right there, they're going to provide a way to modify the information, right? Both going up to your body as well as going down and all around your brain right there. All right, so what happens at the synapse? This is almost exactly, we're going to repeat what we talked about with only a little bit more detail, what we talked about the neuromuscular junction, when we had that motor neuron go to the motor end plate, right, and create that action potential in a muscle fiber, which went down and caused that contraction. That was a synapse, and pretty much the same thing is happening, right? I don't know how much we talked about this step, so we're going to go a little bit further. Now you know what this nerve impulse is. Now you know this is an action potential going down the axon. And remember, action potentials are just a single strength signal, right? They're always the exact same strength. So if there's one signal, one single action potential coming down here, there's a rapid, strong depolarization, right? That is, the cell went from negative to positive. And that depolarization opens up these voltage-gated calcium channels, right? Because remember, those voltage-gated calcium channels, they were closed when the cell was, say, minus 70 millivolts, and maybe, this is just an example, don't worry about it. And maybe they open when an action potential comes down, and all of a sudden you've gone down to, say, I don't know, minus 50 or something, whatever, doesn't matter. There's a certain voltage that it opens up that's caused by this action potential, right? And calcium, which is very much more highly distributed in the extracellular fluid than the inside, because it's going to rush down its concentration gradient. So that's going to flood in there. And instead of causing a depolarization event, which it may or may not, the voltage-gated calcium over here is going to interact with these synaptic vesicles right here, right? But the calcium floods in response to action potential. Okay, so what the calcium does in some magical way that you don't really have to know, it's going to serve as a signal, right? This is a lot of what calcium does when you bring it into there. It causes all sorts of things other than just bring a positive charge in. What it does, though, in this case, is cause the exocytosis of the synaptic vesicles. Those synaptic vesicles are filled with the neurotransmitter. right? The chemical signal, right? They're already there. They're waiting to go. They're already packaged there. All they're waiting for is this calcium signal. And what that calcium signal makes them magically do is fuse with the plasma membrane of the presynaptic membrane and release the contents into the synaptic cleft. Right? Neurotransmitter diffuse across there and bind. So these neurotransmitters, the chemical signal, in this case, well, we're not saying what it is. These little red things over here is the neurotransmitter. When we talked about the neuromuscular junction, that was acetylcholine, right, going across here, and it binds to a receptor. Right? In this case, the binding of that receptor over here is going to bind to this so-called ionotropic channel, right? It's a ligand-gated channel. That's also an ion channel. Okay, diffuses across there and binds to this receptor. The binding of the transmitter molecules to the receptor is going to open up the channels, and whatever channels these are, in this case, it looks like a sodium one. Sodium floods in, right? In this case, it's a positively charged one, so that's going to cause a positive charge over here, the postsynaptic potential, right? And this is where we get back to that whole graded potential, right? The depolarization. And as we'll see, there's also hyperpolarization. If this wasn't sodium, if it was chlorine, it would be hyperpolarization. Now you have a positive charge, right? A graded potential and excitatory one in this case because it's sodium. And if it's strong enough at this place, and if it reaches the trigger zone down near the axon, it will trigger an action potential in the next cell. So that is what happens in neural communication. One cell talking to another cell. The action potential reaches the end of the axon terminals, and the message is passed to the next cell through that synapse. And if that graded potential resulting from that synaptic input is great enough, and it reaches the threshold around the axon hillock, a message will be passed to the next group of cells down that cell's axon. All right, so now you can incorporate all this in together, and next time we'll talk about how the cell integrates various inputs from multiple neurons to coordinate its output. All right, we'll see you next time.